our work about using a synthetic gene circuits to study the quantitative behavior of microRNA-mediated regulation. The majority of the material in this talk has been published last year on PNAS. As you know, microRNA is a class of important regulators in the cell. By sequence complementary to the target's genes, it can direct the risk complex to bind the targets. With imperfect pairing, they will trigger the repression of the translational process and accelerate the degradation of target genes. With perfect pairing, they will trigger directly cleavage of targets, which is also known as the SIR effect or RNA interference. People have found that only 7 base pair complementary to the target's gene, called the seed region of the microRNA, can trigger microRNA target hybridization. So you can imagine that in the human transcriptome, there can be a lot of microRNA binding sites. So each microRNA can bind to dozens, even to hundreds target genes. And each target gene can be bound by multiple microRNAs. So they compose a very complex network. Traditionally, when people talk about microRNA target prediction, they're talking yes or no model. That means that you think the gene is active. When the microRNA is expressed, it is repressed. However, the reality is much more complex. You can imagine that if the targets have very high expression level and the microRNA has low expression level, even though the microRNA can bind in part of the targets, the majority of them will bypass the regulation of microRNA. On the other end, if the microRNA express a lot, all the target genes will be repressed. While in the middle, the system is very sensitive to the subtle changes of either microRNA or target genes. Such kind of quantitative behavior was first started in bacteria for small RNA regulation. They first proposed a threshold model. That means when the small RNA are fixed at a certain expression level, if the transcriptional level of the targets is lower than the threshold, all the target genes will be degraded and there's no translation. If the target is higher than a certain level, part of the targets can be translated into the protein, and you will see this protein expression. Similar phenomenon has been validated in mammalian cell for microRNA regulation. More recently, people found another language for RNA regulation called competing endogenous RNA effect, or CER effect. Let's just take a simple example. We have one microRNA species with 2 target species. The transcriptional level of the targets 1 and the microRNA do not change, but the targets 2 have different expression levels in different conditions. So along with the increase of the CER2 RNA level, more microRNAs will be titrated and spawned to bind this type of targets, and there will be less microRNA available to bind the first targets. So both of these genes will have upregulated their translational level, and the gene's translation is de-repressed by this competing endogenous RNA. So this phenomenon was thought to be widespread. You can imagine in the cell, there's a large pool of microRNAs. There are many kinds of targets competing with this pool, like the mRNAs, link RNAs, pseudogenes, and more recently, the circular RNAs. So it's a very complicated and quantitative process. People want to understand the quantitative behavior of this model. It's not hard to write down this simple model into a kinetic model to describe this process. For example, this one represents the CER1. It has certain rate to generate the free mRNA. The mRNA can be degraded, translated into protein. This one represents the microRNA gene. It also has certain kinetic rate to generate the mature microRNA. The microRNA can bind to the free mRNA species as a complex to accelerate the degradation process. The lower part is symmetric to the upper part, so it's just 2 targets and 1 microRNA. You can also expand it to multiple microRNA and a multiple target system. You can directly write down ordinary differential equation to describe this model. However, as you can see that, this model is nonlinear, and most of the parameters in the model is very hard to be measured directly. So it's hard to understand the kinetic parameters of the system. In our work, we try to turn the help of synthetic biology. We translate this model into a synthetic gene circuits. In this synthetic gene circuits, we've used 4 different fluorescence proteins. In the IFP gene, we put a microRNA in the intron of this fluorescence protein. Along with the transcription and splicing of this gene, the microRNA will be produced. We put 2 target genes in the system. One is MCAT, it's driven by a constitutive promoter. The other one is UIFP. It is regulated by DOX. Without adding the DOX, there are 1 microRNA, 1 target system. When we add DOX, the UIFP will express and compete with MCAT for the microRNA binding. And the TAC-BFP is driven by a constitutive promoter as a control of the system. Then we can use flow cytometer to monitor the intensity of these fluorescent proteins in each single cell and get the intensities. Next, we try to put our model and the synthetic gene circuits into the computer and human hex cell line, respectively, and we can play with the model and change the synthetic gene circuits, respectively, help us to understand the behavior. We have observed several interesting phenomena. First, we asked what factors can influence the strength of CER effect. How about the microRNA expression level? How about target expression? How about the microRNA binding sites number and the microRNA binding sites affinity? First of all, our model predicted that microRNA abundance and target binding strengths can both influence the CER effect. The x-axis is the transcriptional level of the CER1, and the y-axis is the protein level. The red curve is the system without adding the competing endogenous RNA, and the blue one is with the competing effect. So you can see that the blue curve is always upper of the red curve because the gene is de-repressed by the competing effect. The larger the difference between these 2 curves, the stronger the CER effect. You can see that along with the increase of the microRNA level, the difference between these 2 curves become larger and larger. And the region with the maximum de-repression effect shifted to the higher transcriptional level of CER1. The binding strengths also influence the competing effect. However, they do not influence the position of this effect. They just influence the intensity of the CER effect. So we tested this hypothesis by our synthetic gene circuits. We put different amount of the microRNA into the system. As you can see, if the microRNA expressed very low, the de-repression effect is very subtle. If the microRNA is too much, you also cannot see such kind of de-repression effect because the transcription of the targets cannot reach the region theoretically can achieve the maximum de-repression. Only in the middle you can see the best effect. We also put different number of target sites on the 3' UTR of the target genes, for example, 0, 1, 4, 8, 12. The more sites, the stronger the binding and the stronger the competing effect. And we try to put different target site types into 3' UTR from low binding affinity to strong binding affinity. And you can see that with the stronger binding affinity, the stronger the CER effect. Another interesting phenomenon we observed is about the ultrasensitivity of the system. So this is the dose-response curve of microRNA regulation. The y-axis is the protein level of targets. The x-axis is the microRNA expression level. The red curve is the targets without competing effect. You can see it's kind of flat. When we add more competing endogenous RNAs into the system, the curve shifted to the right, and it's become sharper and sharper. So such kind of competing effect can introduce ultrasensitivity into microRNA regulation. Another question we are interested was about the crosstalk between the RNA pathway and the microRNA regulation. As you know, when we design siRNAs, we design perfect complementary small RNAs that can trigger the direct cleavage of the target gene. However, some of these small RNAs may bind to the other genes through the microRNA pathway, say the seed match. This is called the off-target effect for siRNA application. And this is a major problem for many cases for siRNA design. So we built a very specific synthetic circuits to study the system. We keep the AMP kit with the micron type binding sites in the 3' UTR, so it's with a barge. It will go to the micron pathway. In the EYFP, we put perfectly complementary target sites, so it will mimic the target of SIR effect. When we look at the dose response curve of the micro repression, we saw that these two kinds of targets have very different dose response curves. So if you have very low micro level, both of them will be not repressed. If you have too much micro or SIR, both of them will be repressed. Ideally, you should find a certain amount of SIR that can efficiently repress your targets without touching your off-target. So we introduced a curve, we call it on-target, off-target repression phase plan. So at a certain SIR level, you will have two cross points with dose response curves. We use those two values as the x-axis and the y-axis of our plan. So when you have SIR level from low to high, you will find a curve on this phase plan to describe the relative repression efficiency between the target and off-target. So for different target and off-target pairs, you will find the curves with different shapes. For good SIR design, the curve will close to the upper left corner because at this region, you can efficiently repress your primary target and have less effect on your off-target. So we asked what factors may influence the relative repression efficiency. How about the relative dose between the target and off-target RNAs? And how about the binding affinity of these stars? Interestingly, we found that the relative expression of the targets and off-target doesn't matter. This has been also validated by our synthetic circuits. That means that if you want the system to work on this point, that means you have a certain amount of repression of your target and off-target. If your off-target genes express a lot, they may titrate some microRNA out, and the point may go to the purple one. But you can add more SI into the system to compensate this effect and move it along the curve to this point. However, the binding affinity greatly influences the shape of the curve. That means that if your off-target have very strong binding affinity, when you repress your target, you will also have very strong off-target effect. Theoretically, we have proved that only the free microRNA is essential for the whole competing system. So this is the free microRNA. This is the one bind to the targets. This one is essential in the equation that can influence all these targets and off-targets. So when you study the SIR with multiple targets genes, actually, they always work at the same point at the x-axis. So for each off-targets, you may have a curve, and all of them will work on the same point on the x-axis. And you can study each of the off-targets effect using the system. The concluding mark is that, previously people are talking about microRNA targets prediction. We call it old generation. It's just to predict yes or no, whether a microRNA can bind to their target or not. However, people are finding that the microRNA regulation are highly quantitative dependent. And you can imagine that in the cell, there's a very complex system composed by different types of RNAs, link RNAs, and circular RNAs. They all can compete with the microRNA and titrate the expression level of each other. Here's the knowledge. This work is done by my students Ye and Xinying, and it's under close collaboration with my colleague Zheng and his students Bing Liu, and the founding resource. Thank you very much.